All right, welcome to part 4 of the Mod 13 lecture videos on genetics. In part 4, we'll be talking about the chromosomal basis of inheritance and sex-linked traits and genes. So chromosome behavior accounts for Mendel's laws. However, when Mendel was doing his experiments, scientists did not have an understanding of what happened during mitosis and meiosis yet. They actually didn't really know the specifics of these two processes until a long time after Mendel's death. And then once they figured out the specifics of meiosis, they started to realize that, hey, the behavior of the chromosomes in meiosis can be explained by the laws that Mendel proposed years and years before. So the chromosome theory of inheritance holds that genes occupy specific loci or specific positions on chromosomes, and those chromosomes undergo segregation and independent assortment during meiosis. Mendel's laws correlate with chromosome orientation and separation in meiosis. So his two laws, the law of independent assortment and the law of segregation, they explain the behavior that we see the chromosomes do during meiosis. So the law of independent assortment explains the orientation of homologous chromosome pairs in metaphase 1. Remember, the homologous chromosome pairs can assort independently or assort randomly. So it doesn't matter which one's on the left and which one's on the right. It's completely random. That's what the law of independent assortment refers to. And then the law of segregation refers to the separation of homologous chromosomes in anaphase 1, which results in only one chromosome going to each gamete. And if there is a loci on one chromosome carrying one allele, that means that each gamete will only receive one copy of the allele, which would result in a haploid cell. We can see this better as we follow this F1 generation going through meiosis. So the F1 generation, this F1 generation is all yellow round seeds that are heterozygotes, so capital R, lowercase r, capital Y, lowercase y. We can see the R and the Y chromosomes here. The R chromosomes are slightly longer than the Y chromosomes. And again, the maternal chromosomes in red, the paternal chromosomes are in blue. The first law of independent assortment states that the chromosomes orientate independently at the metaphase plate. And so we can see there's two equally probable arrangements of chromosomes at metaphase 1. We can either have all the maternal chromosomes on the left, or we can have kind of flip-flopped, so the lowercase r, capital Y, lowercase y, compared to over here we have capital R, lowercase r, and then capital Y, lowercase y. So either one of these orientations is equally probable. And then as we go to the next step, we have anaphase 1. Remember, anaphase 1 is when the homologous chromosomes separate. So this chromosome is going to the left side of the cell along with this one. And then this lowercase r and this lowercase y are going to the right sides of the cell. Over here in this orientation, this lowercase r is going to the left and the capital Y is going to the left. And then the capital R and the lowercase y are going to the right. As a result of this, we see these chromosomes lined up at metaphase 2, and then this represents meiosis 2, and we see these gametes formed from these different orientations of the homologous chromosomes at metaphase 1. So this bracket shows meiosis 1, this bracket shows meiosis 2. For simplicity, not all the steps of meiosis 1 and meiosis 2 are shown in this figure. If you're confused at all about the specific steps, refer back to the cell division lecture. But the purpose of this figure is just to illustrate that these are all the possible gamete combinations from this F1 generation. We're pretending that it just has 2 chromosomes. Typically, Ps have 7 chromosomes, but to simplify this figure, again, we're just showing the 2 chromosomes, the long R chromosomes and the shorter Y chromosomes. So if the chromosomes orientate this way at metaphase 1 with the reds on the left and the blues on the right, and then go through the remainder of meiosis 1 and meiosis 2, we'll end up with 2 gametes that are capital R, capital Y, and then 2 gametes that are lowercase y, lowercase r. If the chromosomes orientate this way during metaphase 1, which is equally probable as them orientating this way, and then proceed to go through meiosis 1 and meiosis 2, we will have 2 gametes with capital Y lowercase r, and then 2 gametes with capital R lowercase y, capital R lowercase y. These numbers, there's 1, 2, 3, 4, 5, 6, 7, 8 total gametes, and then each one of these is 2, so 2 eighths, 2 eighths, 2 eighths, and 2 eighths. That reduces down to 1/4, which is how they got the 1/4. So 1/4 of the gametes will have capital R, capital Y. 1/4 of the gametes will have lowercase r, lowercase y. 1/4 of the gametes will have lowercase r, capital Y. And 1/4 of the gametes will have capital R, capital Y. These proportions down here are actually what we use to create the Punnett square. So take a look at all of these, and then on the next slide, I'm going to show you the Punnett square. Note that each 1/4 proportion or percentage of the gamete gets a spot on the Punnett square. So 1/4 capital R, capital Y, 1/4 lowercase r, capital Y, 1/4 capital R, lowercase y, and then 1/4 lowercase r, lowercase y. Those are the exact same frequencies that we got when we followed the F1 generation through meiosis 1 and meiosis 2. And that's what we put on our Punnett square. We've briefly looked at this Punnett square before when we were talking about independent assortment and when we were talking about dihybrid crosses, because this is a dihybrid cross. And notice the eggs and the sperm, they are both from that F1 generation, so they both have the exact same genotypes. So the proportions or the percentages are the same. So 1/4 capital R, capital Y, 1/4 lowercase r, capital Y, 1/4 capital R, lowercase y, and 1/4 lowercase r, lowercase y. If you were to then fill out this Punnett square, you would get the stereotypical phenotypic ratio for a dihybrid cross, which is 9 to 3 to 3 to 1. 9 yellow and round to 3 green round to 3 yellow and wrinkled to 1 green and wrinkled. So Mendel's laws explain the movements of the chromosomes that we see in meiosis. That's the big takeaway from this section. Now we're going to end this lecture talking about sex chromosomes and sex-linked genes. So chromosomes determine sex in many species, including humans. In mammals, a male has XY sex chromosomes and a female has XX. Down here, we have an image of the X and Y chromosome. Notice that the X chromosome is quite a bit bigger than the Y chromosome. However, the X and the Y chromosome, the tips of the X chromosome are homologous with the top part of the Y chromosome, so this is how they're able to behave like homologous chromosomes during meiosis. The Y chromosome, since it's so small, it has genes for the development of testes in males, but it doesn't really have many other genes besides that. An absence of the Y chromosome allows ovaries to develop. So an XY individual would be a male. The Y chromosome has the genes that cue the development of the testes. If the Y chromosome is absent, that would allow ovaries to develop. In addition to these X and Y sex chromosomes, human males and females both have 44 autosomes, which are the non-sex chromosomes, and that gives us a total of 46 chromosomes in humans. Because females are XX and males are XY, this means that males are going to determine the sex of the offspring, because females only have X chromosomes, so the only chromosome they are able to pass on to their offspring is an X chromosome. A female will always pass on an X chromosome. Males have an X chromosome and a Y chromosome, so they can pass on either the X or the Y. It's a 50-50 chance that the X chromosome is passed on or the Y chromosome is passed on. Obviously, if the male passes on his X chromosome, the resulting offspring will be female because the mom will also pass on an X chromosome. If the male passes on his Y chromosome, then the resulting offspring will be male because the mother, the female, will pass on her X chromosome. the male will pass on his Y chromosome. An XY individual is a male. So males determine the sex of the offspring because females will always pass on the X chromosome, whereas males have a 50-50 chance of passing on either their X or their Y chromosome. So a checkpoint question based on this: King Henry VIII of England was quick to blame his wives for bearing him only daughters. Explain how, from a genetic point of view, his thinking was wrong. So we just finished talking about how it's the male or the sperm from the male that determines the sex of the offspring, because a female can only ever pass on an X chromosome. So based on that, the male sperm bears either an X or the Y, thereby determining the sex of the offspring. Henry VIII's wives' eggs always carried an X, so it was actually his sperm that resulted in him only having daughters. It is the sperm that determines whether an offspring is a male or a female. We're going to talk now about sex-linked traits and sex-linked genes, because sex-linked genes exhibit a unique pattern of inheritance different from anything we've talked about so far. So a gene located on either sex chromosome is called a sex-linked gene. The Y chromosome is very small. We saw the picture of it 2 slides ago. It really only carries the genes that code for the formation of the testes. It doesn't have much else on it. So typically sex-linked genes are carried on the X chromosome because the X chromosome is a lot bigger and has genes that aren't related to sex determination on it. So the X chromosome carries many X-linked genes that control traits unrelated to sex. We're going to first talk about an example of eye color in fruit flies, and then we'll talk about a few human examples of sex-linked traits. The inheritance of white eye color in the fruit fly illustrates an X-linked recessive trait. So most fruit flies have this red eye color. The trait that is more common in a population we refer to as the wild type trait. So you might hear me say that. The red eyes are much more common in the population. They're the dominant trait. They're the wild type trait. White eyes, an individual has to be, it's a recessive trait, and the eye color trait is carried on the X chromosome. So the white eye color is a lot rarer in the population. We're going to look at a few examples of crosses of flies with different eye colors. So XR is the wild type trait that codes for red eyes. So this female is a wild type female. She has red eyes, and her genotype is XR. So she only carries the red eye allele. This male over here, notice a male only has one X chromosome, so he only needs one copy of the recessive white eye allele, X_r, in order to exhibit the white eye trait. His other chromosome is a Y chromosome. So that's important to note. In sex-linked traits that are carried on the X chromosome, since males only have one X chromosome, they only need one copy of a recessive trait to express that recessive trait, whereas females who have 2 X chromosomes would need 2 copies of that recessive trait in order for that recessive trait to be expressed. So we're going to cross this wild type red-eyed female with the white-eyed male. We see the female can only pass on the XR gamete, and then the male can either pass on his X_r chromosome or his Y chromosome. Sorry, I misspoke a second ago. The female can only pass on the XR allele on both of her chromosomes because she only has 2 X chromosomes. The male has an X chromosome and a Y chromosome, so can pass on either of those. So the eggs will all be XR. The sperm can either be X_r or Y. So we see all the female offspring from this cross will be carriers of the white eye allele, but they will not express the white eye allele because they still have that one XR, the dominant trait. So the females are carriers of the white eye allele, but they do not have white eyes. And then the male offspring are not carriers. They just have the XR allele from the female. And then they got the Y chromosome from their father. In our next cross, we're going to cross one of these heterozygote females with a wild type red-eyed male. So this male is wild type. He has the X capital R X chromosome, so he has red eyes. And then this female also has red eyes, but one of her X chromosomes is the capital R and one is the lowercase r. So she carries the white eye allele. When we do this cross here, something interesting shows up. So we get the 3-to-1 phenotypic ratio again, which we got a lot in the F1 crosses with Mendel's laws in monohybrid crosses. However, notice that the white-eyed individual is always a male in this case. So the females from this cross will never have white eyes, but 50% of the males from this cross will have the white eyes. So the eggs, the possible eggs from this female are either X capital R or X lowercase r, and then the possible sperm are X capital R and a Y. If we do the Punnett square, this square here is X capital R, X capital R. So this is a wild type, only carries the red eye trait. This male over here, X capital R, Y, wild type, red-eyed male who only carries the red eye allele. This female down here, X lowercase r, X capital R, she is wild type. She has red eyes, but again, she is a carrier of the white-eyed trait. And then this male over here, he received the X lowercase r chromosome from his mother. And so the X lowercase r Y shows that recessive white eye allele. So typically we see sex-linked traits that are carried on the X chromosome show up much more frequently in males than females because females can be carriers, whereas males only have one X chromosome. So if they have the recessive trait, it will be expressed. Our last cross, we are going to cross a heterozygote female who's a carrier of the white eye allele with a white-eyed male. And the results of this cross can be shown down here. So the possible eggs, we have X capital R and then X lowercase r, and then the possible sperm, X lowercase r and a Y. 50% of the offspring of this cross will have red eyes and 50% will have white eyes. Of the white-eyed offspring, 50% will be male, and now 50% will be female. Notice that in order for a female to express the white-eyed trait, she has to have 2 copies of the recessive allele. She has to be X lowercase r, X lowercase r. So it's a lot less likely for a female to express this X-linked recessive trait because that female would need 2 copies of the X chromosome that has the recessive trait. Now we're going to talk about X-linked recessive traits in humans, but first a checkpoint question. So a white-eyed female Drosophila, which is the fruit fly genus, is mated with a red-eyed wild-type male. What result do you predict for the numerous offspring? So we have a white-eyed female with a red-eyed male. So we're going to go to our whiteboard. So the female is white-eyed, and then the male is red-eyed. So we know the male, automatically one of the chromosomes will be Y, and if the male has red eyes, the other chromosome is going to be the X capital R because the red eyes are the dominant trait, it's the wild type allele. If the female has white eyes, that means she has to be X lowercase r, X lowercase r, because we just learned on the previous slide that females need 2 copies of that recessive X-linked allele in order to express the trait. So if we then complete this Punnett square, X lowercase r, X capital R, and then X lowercase r, Y, X lowercase r, X capital R, and then X lowercase r, Y, we see that all of the males from this cross over here will be white-eyed, and then all of the females will be carriers but will have red eyes. So let's see if that's what the checkpoint question response is. All female offspring will be red-eyed but heterozygous or carriers, that's what we found, and all the male offspring will be white-eyed, X lowercase r, Y, that is also what we found. Human sex-linked disorders affect mostly males for the same reason, because females need 2 copies of the recessive X chromosome allele, whereas males only need one for the trait to be expressed because males only have that one X chromosome. The other chromosome is the Y chromosome. Most X-linked human disorders are due to recessive alleles and are therefore seen mostly in males. A male receiving a single X-linked recessive allele from his mother will have the disorder, whereas a female must receive the allele from both parents to be affected. So it's a lot less likely for a female to be affected by a recessive sex-linked disorder than a male. That doesn't mean that females are never affected by these recessive sex-linked disorders, but we do expect to see higher numbers of males affected than females. A very common example of a sex-linked disorder is colorblindness, red-green colorblindness. So that is a recessive sex-linked disorder that we do see show up more in males than females. Another more deleterious disorder is hemophilia. So hemophilia is the disease where someone's blood fails to clot. They're missing some of the blood clotting protein. So even a small cut can prove fatal to someone with hemophilia because the cut will never clot. It will just keep bleeding and keep bleeding. Normally, like if I were to cut myself, if you don't have hemophilia and you were to cut yourself, the blood would clot and clot pretty quickly after, like, let's say you bumped your arm into a piece of wood and it started bleeding. After a few minutes, the blood clots and the bleeding stops. Someone with hemophilia does not have that ability. And hemophilia was really common in the royal families of Europe. So Queen Victoria of England was a carrier for hemophilia. So the carriers are denoted by the circles with the two colors, and then circle indicates female, square indicates male. So Queen Victoria was a carrier. She passed the carrier gene down to her daughter Alice, and then Alice passed the carrier gene down to her daughter Alexandra. Alexandra married Tsar Nicholas II of Russia, and their son Alexis was affected by hemophilia. So he inherited the recessive X chromosome from his mother, even though his father was not affected with the disease at all. All it takes is one chromosome from the mother that carries the disease. So Alexis inherited the disease carrying X chromosome from his mother, and so he was affected with the disease. Again, we see X-linked recessive traits such as hemophilia occur much more frequently in males than females because the males only have one X chromosome, whereas females have two. Our last slide in this lecture is a checkpoint question. Neither Tom nor Sue has hemophilia, but their first son does. If the couple has a second child, what is the probability that he or she will also have the disease? Okay, so neither Tom nor Sue has hemophilia, but their first son does. So this means there's a lot to unpack here, but Tom does not have hemophilia. We're going to use X. Capital H means no hemophilia, and then X lowercase h is the recessive hemophilia allele. Tom doesn't have hemophilia. We know he's a male, so he has a Y chromosome, and he doesn't have hemophilia, so he's going to have the X capital H. However, his son does have hemophilia, so that means he's X lowercase h with a Y. That means he got his Y chromosome from his father. I'm going to see. Sue is her name. I forgot her name. Their son got the Y chromosome from his father, but he must have gotten the X, the recessive hemophilia X, from his mother. However, Sue is not affected by hemophilia, so we know that she must be a heterozygote carrier. So she has one dominant allele X chromosome and then one of those deleterious recessive alleles carrying hemophilia. So their son happened to get the X lowercase h chromosome from Sue. Their second child, they want to figure out the probability that it will have hemophilia. If they had a daughter, there's no chance of her having hemophilia because she would inherit the X capital H from Tom. And so even if she did inherit the X lowercase h from Sue, the capital H would counteract it and she would be a carrier but would not be affected. So their only chance of having another child with hemophilia is to have a male child. We already know that it's a 50-50 chance of a male or female child. So I'm going to put that probability here, 1/2 And that's just the chance of having a male child. We get that from looking at the male chromosome. So Tom has a 50% chance of passing on his Y. That would make the offspring male. And he has a 50% chance of passing on his X chromosome, which would make the offspring female. We then need to figure out the chance of Sue passing on her deleterious lowercase h hemophilia allele. And we're going to multiply that using the rule of multiplication, which we talked about in the last video or the second to last video in genetics. So the rule of multiplication is just to determine the probability of an event occurring that can happen in two different ways. You multiply those percentages. So for Sue, she has 2 X alleles. She has a 50% chance of passing on her normal capital H allele and a 50% chance of passing on the lowercase h hemophilia allele. So this is going to be another 1/2 or 50% chance for passing on her hemophilia allele. We're then just going to multiply those two fractions together. So 1 times 1 is 1, 2 times 2 is 4. That gives us a 1/4 or a 25% chance that their next child will have hemophilia. If we take a look back at our PowerPoint, we see that they got the same answer as us. So the probability that their next child will have the disease is 1/4 because there's a 1/2 chance of a male child, and then you need to multiply that by the 1/2 chance that the male will inherit the mutant X lowercase h from Sue. So this concludes our lecture on genetics. I'll see you guys in module 14 where we will start to talk about molecular genetics.